Hello. Production of heterologous proteins has many applications in recombinant DNA technology. In this session, I am going to talk about how a recombinant DNA can be expressed in a host organism. First, what is recombinant proteins? Recombinant proteins are the proteins which is made by recombinant DNA technology, and it has many applications. So, in recombinant DNA technology, we will be taking a foreign DNA and expressing that protein DNA in another host. We can express a animal protein in a bacterial cell, or we can express a bacterial protein in a plant cell. So such type of manipulations can be done using the recombinant DNA technology, and we can manipulate the protein expression of a host cell. So it has many applications in diagnostic tools. It has applications in vaccine. As we know, most of the vaccine we are using against this coronavirus is recombinant DNA vaccines, just like the Covishield. It's a recombinant DNA vaccine, and it can be used for making therapeutics like the recombinant insulin or the human growth hormone. Then the proteins made by recombinant DNA technology is used in detergents, cosmetics, food production, and food additives. So it has many applications, and when we are using this recombinant DNA technology, it actually provide a efficient way to produce large amount of pure proteins. So if you take the example of growth hormone, in order to produce a small amount of growth hormone from other sources, just like from sheeps or from cadavers, it take lot of time, lot of money, and the production quality is very less. But if you are producing the same protein using Escherichia coli as a host organism, the production process is very easy, and the cost of production is also very low. So it will be more available in a cheaper cost. So we can produce a large amount of protein in a pure form using recombinant DNA technology. Another advantage of recombinant DNA technology for protein production is like we can actually manipulate the properties of the protein. We can humanize the protein, or we can make it more stable. Maybe we can, if it is an enzyme, we can manipulate the enzymatic activity. So at one point, we can actually produce large amount of proteins in a pure form, and at the same time, we can actually manipulate the properties of the protein. So the recombinant DNA technology is getting more and more popular in producing these proteins for large industrial application as well as for the medical applications. So for expressing a protein, we need so many factors, or so many systems. First thing we need is the expression system, which host we should express the protein. We have many choices. So we can express in a eukaryote, maybe express in a prokaryote, or we can express in a plant cell or an animal cell. So we need to choose the best host for the production of the protein. Next, we need an expression vector. Uh, as we discussed earlier, there are cloning vectors and expression vectors. So expression vector is having elements which is actually direct the protein expression of the cloned gene. So we need expression vectors for that. And in the expression vector, we need expression cassette or the sequence for expression, which include the promoter, ribosome binding site, and terminator. Then the codon usage should be optimized, then protein sequence should be optimized, and the protein degradation should be controlled. So we need to do so many things to get a protein expressed. So we are going to consider each point. uh, like the first one is the expression system. As we know, Escherichia coli is the major expression system because we have a extensive knowledge about the molecular biology of the Escherichia coli. We know the promoter sequence, we know the ribosome binding site sequence, we know about the terminators. So, and it is most widely used organism for cloning, and it is also a most widely used organism for expression. And it will grow in simple and inexpensive media. So large scale production is very easy. Then it can downstream processing from the Escherichia coli is also very easy. The harvesting and the purification process is also easy when we are using an Escherichia coli. But Escherichia coli is having some disadvantages. One of the major disadvantages is that it do not have a machinery for processing introns. As we know, the eukaryotes are having introns, but prokaryotes do not have introns. So if you are inserting a eukaryotic gene in a prokaryote or in Escherichia coli, it do not process the introns. So intron processing machinery is not there in the Escherichia coli. That's one disadvantage. Secondly, it do not modify the protein. As we know, after the protein synthesis, there is a post-translational modification in which proteins are extensively modified by different types of different groups like the glycosylation or acetylation or lipoification, things like that. So different groups will be attached to the protein. But in Escherichia coli or the prokaryotes do not do that extensively. So if you are using an Escherichia coli, there is no intron processing mechanism, and if you are using an Escherichia coli, there is no post-translational modifications. More than 95% of the eukaryotic proteins are post-translationally modified, and in order to get the proper activity of the protein should be modified post-translationally. So when we are expressing a eukaryotic protein in a prokaryote, we just miss this post-translational modification. So there are other choices. We can use the yeast cells or insect cell lines. Plant cells or animal cells can be used. These cells have the intron processing machinery as well as the post-translational modification machinery. Or we can use the molecular farming, in which we will use higher eukaryotes for the production of recombinant protein. But when we are using higher eukaryotes, it will become more and more complicated than using an Escherichia coli. Second thing we need is an expression vector. Expression vectors are designed to express the protein but which we are going to clone in that vector. It has the ability to control cells machinery for protein synthesis. So these vectors will have different elements for expression, and the copy number of the vector is very important. The copy number should not be very high. If the copy number of the vector is very high, the protein expression will be reduced, because most of the energy of the cells will be used for the synthesis of the protein, synthesis of the or maintenance of the vector, rather than for the synthesis of the protein. So we should have an optimum number of vectors. If the number of vector is low, the expression level will be low. If the number of vectors is very high, the expression level also drop, because of the energy usage will be more directed for maintaining the plasmid rather than making the protein. So we are using most commonly used origin of replication for expression vectors are from PBR322 or PUC80. This is the general structure of the expression vector, in which you can see the selectable marker, origin of replication, and an expression cassette. In the expression cassette, we have promoter. This is where the expression begins, and it is actually controlling the level of gene expression. And at the other end, we have a terminator, the transcription terminator. In between, we have a ribosome binding site and a multiple cloning site. Whatever gene you are going to clone here will be expressed as a protein, because it is preceded by the promoter and ribosome binding site, and at the end we have a terminator sequence. So this is a general picture, and in the expression cassette we have a promoter. Promoter is where the transcription is initiated, and the promoter is where we are actually controlling the gene expression. And for production of recombinant protein, what we need is a strong inducible promoter. What's a strong and inducible promoter? Strong promoter means it has the ability to produce more amount of mRNA. If a promoter is strong, it will bind to the RNA polymerase very frequently, and it will produce huge amount of mRNA, and this mRNA will produce protein. So we need strong promoter. So we'll get more amount of mRNA, and eventually we will get more amount of protein. Secondly, we need an inducible promoter. Inducible promoter means the promoter expression can be induced in some way. The induction can be maybe tissue specific, or maybe a certain time of development, and most commonly in the presence of an inducer. If the inducer, that's maybe a chemical, or a like maybe a physical condition like heat or presence of light. So if the inducer is present, the promoter will be activated and the protein will be expressed. So for recombinant DNA technology, we need some level of control of gene expression. So we prefer the inducible promoters. Most commonly the promoters which can be induced by physical or chemical factors. And we have two examples, Lac promoter and Tac promoter. The Lac promoter is the one we are very much familiar with. It is the promoter of lactoperon. It is an inducible promoter. It can be induced by lactose or IPTG. IPTG is an analog of lactose. But this promoter, even though it is used in cloning vectors very extensively in the PUC80, you can see this, but it is not really used in other expression vectors. There are different reasons. One thing is that the Lac promoter is fairly weak. It is not a strong promoter. It's a weak promoter. And secondly, it has a property of promoter leakage. That means in the absence of inducer, absence of lactose or the IPTG, it will express in some base levels. So those type of promoters are called leaky promoters. If it is expressing in the even in the absence of inducer, it's called a leaky promoter. Lac promoter is a leaky promoter. Even in the absence of the inducer, it will be expressed in a significant amount. So it is not strong and it is not that much inducible. So we are not using the Lac promoter so extensively. But with the second promoter, the TAC promoter is actually a variant of Lac promoter. It's actually a hybrid promoter. We are making it by combining two different promoters, Lac promoter and TRIP promoter. TRIP promoter is the promoter from the Escherichia coli, tryptophan optron. So we are taking the minus 35 element of tryptophan optron and a minus 10 element of the Lac promoter, and we are combining it together. So when we are comparing, it will become more inducible, like using lactose or IPTG. At the same time, it is become more strong. It is 5 times stronger than the Lac promoter. So the TAC promoter is the combination of tryptophan promoter and the lactose promoter, and it's a pretty strong promoter. Once we put a polypeptide or the gene under the control of TAC promoter, it will accumulate to a level of 20 to 30% of total cell protein. So that's fairly a good amount. This is how we are making the Lac promoter. We are taking the minus 10 element from the Lac copron and minus 35 element from the tryptophan. By combining these two, we are making the TAC promoter. Next, the transcriptional termination. Efficient termination is essential for the RNA stability. If the RNA is more stable, it will last in the cell for more time, and if it is in the cell for more time, it will produce more protein. So we need properly terminated mRNAs. So uh, it increases the mRNA stability and also increase the accumulation of protein. In Escherichia coli, the most commonly used terminators are T1 and T2, which is derived from rRNA-B of rRNA operon of Escherichia coli. In eukaryote, we are using AAU, AAA, which is a transcriptional termination signal as well as a polyadenylation signal. Next thing to be considered during the time of gene expression is the codon usage. As we know, we have different codons. We have 64 codons for 20 amino acids and stop codon. And many codons are coding for the same amino acid. But the thing is that different organisms use different codons preferentially. For example, for glycine, we have 4 different codons. But some of the organisms will prefer one codon, other organism will code the another codon. Even though the genetic code is universal, some organism has preference for some codons, specific codons. So when we are transferring one gene from one organism to another, this organism may have differences in the codon usage. Escherichia coli and humans are having different codon usage. The codon which is commonly used by Escherichia coli for coding glycine is different than the codon we are using in our genome for coding glycine. So when we are transferring a DNA from human being to the Escherichia coli, because of this mismatch in the codon usage, it will long time for the transcription, because we are having the translation machinery is running by tRNAs. So based on the tRNA abundance, we are using the codon. Anyway, if the codons are different, the preferential codons are different, there will be delay in the transcription. In order to avoid that, we have two different methods. One is expression strains. Expression strains like Rosetta, it's actually a recombinant strain. It actually expresses human codons. Because of this recombinant DNAs in it, it has same codon usage as humans. It's a different types of Escherichia coli. Or we can go for codon optimization. Before transferring the gene from the humans to Escherichia coli, we will use the site-directed mutagenesis to convert the codons to the E. coli preferred codons. So that's what we call the codon optimization. So codon usage can be manipulated, or usage can be optimized either by using expression strains or using codon optimization techniques. Next is protein sequence. Once RNA is converted to the protein, what happens is, like, if a protein is found to be foreign by Escherichia coli, it will degrade the protein. So in order to avoid that, we need to have some idea about, we should have some idea about the how the Escherichia coli is degrading the protein. So then we have the end rule of Escherichia coli. If the we know that proteins are having N-terminal and C-terminal. In the N-terminal, the amino acid present in the N-terminal is actually a major factor which is deciding the protein degradation. If the N-terminal of a protein is having amino acids like arginine, lysine, leucine, phenylalanine, tryptophan, and the tyrosine, if these amino acids are present in the N-terminal, what happens is like the protein will be degraded very quickly. If these amino acids present, 50% of proteins will be degraded within 2 minutes of translation. If any other amino acids are present at the N-terminal, the protein will have more stability. Its half-life will be 10 hours. So if you are using any of these amino acids, the specified amino acids, the half-life will be 2 minutes. If you are using anything other than that, the half-life will be increased. So that's how we can manipulate the protein sequence. And even though we like escape from the protein degradation by the N-terminal rule, there are some other mechanism to remove the foreign proteins. Because when we are expressing a heterologous protein in a different host, this protein may not be soluble. It may aggregate or it may precipitate. Once the aggregation or precipitation happens, the cell machinery will clear off that protein using protease enzyme. So if the abnormal or misfolded proteins happens in a cell, it has innate mechanism to degrade that. To avoid the degradation of recombinant protein, we can use different strategies, which includes use of protease deficient Escherichia coli, the Escherichia coli which is mutated to not to express the protease enzyme, or we can grow the cells in low temperature. If you are growing in a low temperature, the metabolic rate will be less and the protein degradation will be also less. Or we can use fusion proteins. We can fuse our protein with some of the Escherichia coli protein, so the protein will be more stable than the normal protein. Or we can target the protein to the periplasm. Periplasm is the region between the cell wall and the cell membrane. So the periplasm is having less number of proteases. So if you are targeting the protein to the periplasm, it will be isolated from the protein degradation machinery of the Escherichia coli. So this is another figure of PGX123. It's an expression vector. You can actually see different regions in this expression vector. So most commonly used expression system is Escherichia coli, but sometimes we use yeast for expression. And there are unique advantages for using yeast expression system. One thing is the post-translational modification, and along with that is just a single-celled organism we can manipulate as like Escherichia coli. It grow fast, it use simple medium, and you live in inexpensive medium, and generally give high expression levels, just like the unicellular prokaryotes. The unicellular eukaryote have higher productivity than other eukaryotes, high yield. Then it, it can process the introns because it is eukaryote, and it is more stable than other eukaryotes. Then production cost and purification cost is less when we are using yeast. And most commonly used yeast is Saccharomyces cerevisiae. It's a unicellular yeast, so unicellular fungi, and it is very much similar to Escherichia coli, except that it's a eukaryote and it is having a nucleus. And it is having most of the post-translation modifications of other eukaryotes. In Saccharomyces cerevisiae, either we can use constitutive promoters like phosphoglycerate kinase, PGK, or GDP, or ADH1. But what we prefer is the inducible promoters, just like GAL system. The GAL system, for a brief introduction, the GAL system is a part of Lillorir pathway, the pathway which is actually converting galactose into the glucose 6-phosphate. But once it becomes glucose 6-phosphate, it will be involved in the glycolysis. So it's a galactose degradation pathway, and GAL genes are expressed in the presence of galactose. When galactose is a sole carbon source, it will be expressed. And GAL1 gene is actually coding galactokinase, and which is under the control of a transcriptional activator which is called GAL4P. And we can actually clone this GAL1 gene, have a promoter called a GAL1 promoter. For expression vectors, we will clone the GAL1 gene under GAL1 promoter, also the GAL1-4P also under the GAL1 promoter. So both these genes are actually the activator, and the effector proteins will be cloned under the same promoter. So here we can see we have the PGAL1, which is a promoter for GAL1 promoter, and it's controlling gal4, which is activator. Once it is made, it will go and bind to this gal1p promoter and increasing the expression. So we can actually increase the expression manifold. So that's about the expression system. Thank you very much for watching.